Okay, welcome to the final lecture of the session. Wow, seems like it's gone a bit fast. I don't know if that's all your experience, but it's definitely mine. So today, I just want to go over basically the wrap-up and key concepts from each of the 10 lectures that I gave in here. And starting with the big picture lecture, we talked about atmospheric CO2 concentrations going up, which I'm not showing you, but we also talked about some of the carbon sinks and carbon sources globally, paying particular attention to this green bit here, which is photosynthesis, or the land carbon sink, because this is where plants are involved. This is where plants are taking CO2 out of the atmosphere. Notice that there can be quite a lot of variability in the land carbon sink from year to year. And then we have carbon emissions such as fuel and industry, biological respiration, and disturbance. Disturbance is shown here as land use change. Then we moved on to we mentioned stomata. I'm not going to cover that too much because we had a whole lecture on stomata. But essentially, we talked about stomata being the link between the carbon and water cycles because of vegetation, and that Australian photosynthetic productivity is related to variability in climate and precipitation. What's special about Australia is that it has the smallest discharge of water to the ocean from rivers, and it also has the smallest annual amount of rainfall of any other continent on the planet. We also talked about how transpiration and stomatal conductance respond to various driving factors, the three most important being radiation, being light, vapor pressure deficit, so here is solar radiation, vapor pressure deficit, and soil water content. And we talked about how vapor pressure deficit has this two different ways that it affects transpiration. When vapor pressure deficit is low and you increase vapor pressure deficit, transpiration increases really quite quickly. But then when you start to get to high vapor pressure deficits, stomata start to close faster than evaporation is increasing. And then you see a decline in transpiration and stomatal conductance. And notice that you need to have a sufficient amount of soil water available before you can really expect a plant to be physiologically active. So then we moved on to light and photosynthesis. Two things about light and photosynthesis. When chlorophyll absorbs light in the photosynthetically active radiation range, so 400 to 700 nanometers, the visible light, but it's especially good at, chlorophyll is especially good at absorbing light in the blue and the red spectrum. And then chlorophyll can also emit a bit of light as a form of photoprotection, and this is what we call fluorescence, and it's just slightly more red or slightly closer to infrared than what the leaf absorbs. One of the main themes across the entire subject is leaf thickness. Leaf thickness relates to a lot of different things regarding plant physiology. And so the structure of a leaf is you have these palisade cells. Remember, the palisade cells are really efficient at absorbing direct sunlight. So when you have a leaf that's in direct sunlight, they will have 2, 3, maybe even 4 layers of these palisade cells. So if you have 2, 3, or 4 layers of these palisade cells, you're also going to have a much thicker leaf. And this is how you can determine whether a sunleaf is thicker or a shade leaf is thicker. A sunleaf will be thicker because it has all of these columns of palisade cells and to grab up all of that extra light.